So as I mentioned before, that in case of rheumatoid uh, arthritis, there is an inflammation in the joint, and it is actually, there should be a trigger in case of rheumatoid arthritis. The trigger that predisposes the inflammation in the joint may be infection, may be inflammation, may be trauma. So now the condition becomes abnormal. So under abnormal condition, the cells, for example, T helper 1, T helper 2, macrophages, and B cell, they can enter the joint. However, in normal condition, it is strictly forbidden for cells to come and settle and sit here or enter the joint. But under abnormal condition, following, for example, trauma or severe infections, so cells, they come and sit here. Normally, these immune cells, they die by apoptosis. And in a normal condition, apoptotic cell, when they die, these cells, they are removed by a special subset of macrophages. But however, when these cells enter the joint, and when they die by apoptosis, the apoptotic cell and necrotic cell, they cannot be removed by that subset of macrophages due to absence of that subset of the macrophages. And during apoptosis, the formation of some proteins known as citrullinated proteins. So during apoptosis, citrullation of some proteins is actually a physiological process. And this citrullation, it means that we remove a molecule of arginine, so the protein, it becomes negatively charged. And so citrullation of proteins, this is a physiological process, takes place during apoptosis or during death of these cells. So when these cells come and sit here and accumulate it here, and due to absence of that subset of cells that have the capacity to clear or to engulf them, citrullinated proteins will get accumulated in the joint. Actually, these antigens or citrullinated proteins will be taken by the antigen presenting cell macrophage, and then get processed enzymes in, and then at the endoplasmic reticulum, they will bind to major histocompatibility class 2, and then the citrullinated peptides get expressed on the surface of the macrophage in association with major histocompatibility class 2. And as can be seen here now, T helper 1 will interfere, because as you know that T helper 1 is a blind cell. T helper 1 cell has no capacity to see tertiary protein, but they can see processed or primary protein. So T helper 1 will interact with presented peptide by the macrophage. This is citrullinated peptide, then get activated. When T helper 1 get activated, they produce some cytokines, for example, interleukin number 2 and interferon gamma. Interferon gamma recruit more macrophages, and the recruited macrophages produce another some cytokines, tumor, for example, tumor necrosis factor alpha and interleukin number 1. Actually, this story, it takes place here in this small joint. So Cytokines by the macrophage, human necrosis factor alpha and interleukin. One, they are pro-inflammatory cytokine, and they have the capacity to induce destruction in this area and inflammation. And as I mentioned, that T helper 1 produces another cytokine known as interleukin number 2. Interleukin number 2 activates B cell and T helper 2, so they enter now the game. And actually, the B cell here inside or in the joint, as you know, that B cell is an antigen presenting cell, and also it has a capacity to take citrullinated peptides. So, but however, B cell, they talk only, or they like, there is a crosstalk only, they talk only to T helper 2. And as can be seen here, macrophages, they have crosstalk with T helper 1. So B cell now interact or process the antigen and present it in association with major histocompatibility class 2 to the T helper number 2. And this get to activation of T helper number 2, and activated T helper number 2 produce some cytokines, for example, interleukin number 4, which is a growth factor for B cell, and so B cell get activated and differentiate into B cell memory cell and plasma cell. So plasma cells, they produce antibodies to citrullinated peptides. These antibodies are known as anti-CCB antibodies. So when anti-CCB antibodies encounter citrullinated proteins here due to failure of clearance of apoptotic and necrotic cells, so this will lead to formation of immune complex. And here, as can be seen here, the immune complex is formed from anti-CCB or anticyclic citrullinated peptide antibodies. So when they encounter these citrullinated proteins, they bind to it, and this will lead to activation of the immune complex. This will lead to formation of the immune complex, and the formation of immune complex will lead to activation of the complement component, and this activation actually starts by activating complement component number 1. So this following that complement component number 4 and 2 get activated, and then complement component number 3 get activated, and then complement component number 5 up to complement component number 9. And as can be seen here, the activation of the complement component is not in order. For example, here this is C1 get activated complement, then C4, 2, 3. 3 comes after 4 and 2, and this is actually it is due to discovery of these molecules. So here this is a story of the evolution of the rheumatoid arthritis due to activation of the complement by peptides, by proteins known as citrullinated peptides. And as I mentioned, that citrullination is a normal physiological process during apoptosis. cell apoptosis, and actually the citrullation takes place by removal a molecule of arginine, and this will lead to negatively charged proteins. But however, as the citrullation takes place here inside this area and this inflamed area, and due to failure of the clearance of the citrullinated proteins and the apoptotic cells and the necrotic cells, these cells get interacted here, and this will result in formation of antibodies to citrullinated peptides, and these are known as anti-cyclic citrullinated peptides, or antibodies. However, anti-citrullinated or anticyclic citrullinated peptides, these antibodies, so the test or detection of these could be in early stage of rheumatoid arthritis. So these antibodies, they have predictive value even before appearance of symptoms. And also these antibodies, I mean anti-CCP antibodies, they have diagnostic value, and also it has a prognostic value. So you can follow the efficacy of the treatment by detecting these antibodies every then and now. And also it has been mentioned that some individuals may produce antibodies of IgM against their IgE. So here's IgE, it acts on antigen, while this IgM is acts on antibody. So when they interact, this will lead to formation of immune complex, and immune complex will activate C1, then C4 to 5 up to C9, and this will lead to formation also of anaphylotoxins, and these anaphylotoxins will lead to inflammation here, and this will lead to rheumatoid arthritis, as can be seen here, characterized by hyperplasia of the membrane, destruction of the bone, synovitis, maybe tendinitis, and destruction together with swelling and hyperplasia of the synovial membrane. Thank you very much, Professor Ahmed Boulad.